Hello boys and girls, welcome back for another lesson of biology. Today we are going through lesson 29 of chapter 27, the gene mutation part 2. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. From the last lesson, we know that there are 4 types of gene mutation. There are insertion, deletion, substitution, and inversion. And among the 4 of them, deletion and insertion of base will affect the protein produced a lot more than the substitution and inversion, because in base insertion or deletion, the reading frame of the DNA is shifted, which will lead to a change in amino acid sequence after the point of mutation. With a different amino acid sequence, it will affect the folding to form a protein with 3-dimensional structure. This is the reason why deletion and insertion of base will affect the production of protein a lot more greatly than base substitution and inversion. In this lesson, we are going to give you some examples of gene mutations, such as sickle cell anemia, 廉狀細胞性貧血. It is caused by a base substitution. On the diagram below, it shows you the normal triplet code, CTT, and a mutated triplet code, CAT. One of the base T is changed to A after mutation. After the mutation, a change in the base from T to A will lead to a change in the triplet code in the DNA template. This leads to a change in the mRNA codon, and this is going to code for a different amino acid in the polypeptide. After the base substitution, there is a change in one amino acid, leading to a formation of abnormal hemoglobin 血紅蛋白 molecules inside the red blood cell. With a abnormal hemoglobin, when the oxygen levels is low, the abnormal hemoglobin they will form long fibers, and this leads to a sickle-shaped 鐮刀狀嘅 red blood cell. Normally, for a hemoglobin, they should look like a ball shape. However, when there is a substitution of base, it leads to a change in amino acid in the polypeptide. This is going to lead to another folding shape. And after folding, the hemoglobin becomes a long fiber shape instead of a ball shape. After the base substitution, the hemoglobin changed from a ball shape to become long fibers. And this is going to affect the shape of the red blood cells. A healthy normal red blood cell should be biconcave disc shape. However, after the base substitution gene mutation, now the red blood cells they become sickle shape. This sickle-shaped red blood cell is going to block the blood vessels and damage organs because you cut off the blood supply. After knowing about some examples of gene mutations, such as the sickle cell anemia, which is caused by the base substitution, now let me ask you a question. Does all mutations, such as the gene mutation, always lead to a change in the amino acid sequence? Give you 5 seconds to think. The answer is nope, not really. Not a must in a base substitution. This is because if after the base substitution, the triplet code it is degenerate, it still code for the same amino acid, then nothing change. It will not change the amino acid sequence. So the idea is, if the triplet codes they are degenerate, it can still code for the same amino acid. So no change in the amino acid sequence. That's it for the part of gene mutation part 2. I hope you enjoyed the video. I will see you in the next lesson. Bye bye.